Thank you very much. I'd like to thank the organizers for inviting me here. And so these are my disclosures. So I am an employee of Replimune, and at the end I will talk a little bit about what we're doing at Replimune. So the field of immunology has, of course, transformed the landscape for cancer patients, as is evident by the growth of this and other meetings on this topic. And where a lot of the work has focused recently is that when you can get T cells into the tumor, then many of the immunotherapeutic approaches are quite effective. But many patients just don't have T cells in the tumor. And we're just beginning to understand how that actually happens. And so the goal of many of the things we're doing in immunotherapy today are to really try to get these so-called cold tumors to become hot and get the T cells there. And I think that is one of the main things that oncolytic viruses are really very well designed to do. And they offer a number of important properties that really help with this. So first of all, they are selectively cytotoxic to tumor cells, and I think we're only at the very beginning of understanding the importance of knowing how the tumor cells are dying, and I think this will lead to ways that we can really improve upon that. And I hope to show you some preliminary data we have today that focusing on that may have really important therapeutic implications. Secondly, in and of themselves, viruses have been dealing with the immune system for really thousands of years, and we can take advantage of that. I think those comments at the beginning were very appropriate, and this is a particularly good way to get this process started. And something that we often don't talk about is, given some of the horrendous things we actually do to cancer patients, these are very well tolerated as a class. The side effects we can get into great detail about are really pretty minimal. And I've been struck with some of my patients that come from afar to see me. We go out to dinner the night they're treated. These side effects are very manageable. It's a really important point. And lastly, some viruses, you can use them to express various transgenes. So you can begin to manipulate the system even further. And again, I think we're at the forefront of that. And while T-VEC utilized GM-CSF as maybe the first generation of oncolytic virus, I think the future generations will probably encode other things. And I'll show you again at the end some of the interesting things that one could contemplate. So for me, it really did begin with T-VEC and my relationship with Rob Kaufman, who developed T-VEC out of a company called BioVex. And this is an HSV1 vector. It is deleted in the ICP 34, 5, and 47 genes, and is modified to also encode 2 copies of human GM-CSF. This went through a randomized clinical trial in which we found a 26% objective response rate and 16% durable response rate in patients with advanced melanoma. This led to the approval of T-VEC in the United States, in Europe, and Australia. And I want to show you this because we actually are just about to publish an update of the overall survival in this study. Before I get to that, I'll just tell you that we've recently looked at T-VEC, and we've shown that it does induce immunogenic cell death as you would anticipate. This is data we just published about 2 weeks ago. So we showed that you produce HMGB1, ATP, and ectocalreticulin with the virus. And this was the original overall survival curve, and I heard at the session this morning that we just missed statistical significance with a p-value of 0.051. Nonetheless, every clinical parameter we looked at was positive. The drug was approved, but the plan was always to follow these patients out long-term for survival. And now that we've done that, we do have an update that will be coming out, and the durable response rate has actually increased from 16 to 19% compared to 1.4% in the GM-CSF arm. And the objective response rate went up from 26% to 31.5%. And again, it's an important point that oncolytic viruses, at least with T-VEC, do seem to result in delayed responses, as has been seen with ipilimumab and other immune checkpoint inhibitors. And so the updated overall survival curve is now statistically significant. So the p-value dropped to 0.04, below 0.05. and this is now with median follow-up to 49 months in the intention to treat population. And then in the stage 3/4 M1A population, which on subset analysis did show a very significant improvement in survival, that improvement continues. You can see the tail of the curve, and this was the basis of the approval in Europe in this particular population. So now that we have longer-term follow-up, I think we can say that T-VEC is therapeutically useful agent, and so we've now turned our attention to really understanding how this happens and how can we improve upon the responses that we've seen. So one of the things that I mentioned is that the oncolytic viruses are pretty good at turning these cold tumors hot. And so there's some work that's been done by Tom Gajewski and Glenn Barber and others that have really suggested, at least in melanoma models, that one of the reasons that you get T cells in the melanoma in the first place is due to STING. And STING is a DNA viral sensor, and so the hypothesis is that because of the abnormal DNA accumulating in melanoma cells, that it will trigger STING, and STING can then begin an innate immune response which leads to chemokine release and then recruitment of lymphocytes. So we began to wonder if STING might also be playing a role with T-VEC, and many of the DNA viruses are actually known to phosphorylate AKT1, which can then, in fact, inhibit C-gas and really blocks the STING pathway. This allows the virus to continue to replicate, and we wondered if there might be some relationship then between the status of STING and the ability of the viruses to work. So to address this, we actually found a number of human melanoma cell lines which are listed here, and we actually measured STING levels, and what we found was there was really quite a contrast with the SK-MEL5 line having practically no STING that we could detect to the LOX4M line, which had abundant levels of STING. And we found that when we infected them with the HSV, most of the lines that had low STING were highly susceptible to oncolysis, but the LOX4 line, where STING was very high, was fairly resistant to it. And we went further to show that STING was potentially mediating the lytic ability of T-VEC by making knockouts. And if you can read our paper that came out 2 weeks ago, we went on and did CRISPR knockouts, and we basically showed that we could reintroduce T-VEC oncolytic capabilities by knocking out STING in the cell lines. So we wanted to then take a look in vivo at this, and so we looked at some of the commonly used murine tumor lines, which are shown here, and looked at STING levels, and it's kind of interesting where we see very high STING levels in B16 and CT26, but we actually see very low levels in a spontaneous melanoma line called D4M, which we obtained from Dave Mullins at Dartmouth. And so this is a line that actually is not very responsive to immunotherapy. So this is the D4M line, either mock treated or with an anti-PD-1, which doesn't do much in this, probably because STING is low. And we show that if you treat this with a modified T-VEC, so this is a T-VEC that actually encodes murine GM-CSF, we actually began to see activity suggesting that in these cold tumors that do not respond to immunotherapy, we could really overcome them when STING is low by using a DNA oncolytic virus like T-VEC. And in fact, this also led to improved survival in this particular difficult to treat tumor. So understanding that, we also then wanted to take a look at whether we could improve this response by looking at other agents that are known to affect intracellular pathways, and being in melanoma, we were particularly interested in the MAP kinase pathway because we actually have now up to 6 different agents targeting BRAF and MEK that are already approved for the treatment of melanoma. And when we looked at the cell lines that we had a range of BRAF mutation status, so we began our studies by looking at BRAF inhibitors, and this is just in vitro work using the SK-MEL28, which is a human melanoma cell line that has a BRAF V600E mutation. And what we found was that if you pre-treat the cells with, in this case, Vemurafinib, a BRAF inhibitor, we could actually improve the lytic capability of the T-VEC, which was not surprising. The SK-MEL5 line, which is the one I showed you that has this very low STING level, was also very susceptible to the combination. And not surprisingly, when we tested a BRAF wild-type cell line, the SKMEL2, there really was not an impact of treatment with Vemurafinib. And the truth is, the way the story goes is my grad student did this, and we thought this was interesting, and I said, just as a control, why don't you try a MEK inhibitor and see what happens with that? And that turned out to be a much more interesting story. And so what we found was, again, not surprisingly, in the BRAF mutated cell lines, we actually saw, again, an improvement in the oncolytic capability with the combination, and here's another cell line. But what surprised us was in the BRAF wild-type line, we also saw a significant improvement in the lytic activity, and this was associated with increase in viral replication, which is shown at the bottom in all of these cases. We also tested the D4M line with this in this system. This is the Murine line, which we then went on to use in some of our subsequent studies, and we saw a very similar pattern here, where there seemed to be a very strong additive effect of the MEK inhibitor. This is actually shown with trametinib, but we've actually done this with several other MEK inhibitors, and this seemed to be a general property that when MEK is inhibited, we could improve the viral replicative abilities and increase the lysis of the cells. So we then wanted to test this in vivo, so we started using a human xenograft model, the SKMEL28 line, and what you can see here is that while both monotherapies, both Murine, T-VEC, and trametinib actually decreased the tumor growth rate, the combination led to complete regression of tumors in this system. And here you can see Ki67 staining showing that there's fewer proliferating cells. And this just shows the increase in viral accumulation that occurs. You can see the T-VEC alone compared to the combination. There's much more intense staining, consistent with the fact that there was a higher level of virus being replicated. We also looked at the impact on apoptosis, and we were in fact able to show that there was an increased apoptotic capability with the combination. We know MEK inhibitor is able to induce apoptosis, and we see this by a decrease in phosphorylation of ERK1 and 2, and also an increase in cleaved caspase 3. So next we really wanted to look at an immune competent model to get away from the xenograft, and that's why we use this D4M line. This is a spontaneous melanoma line that grows in C57 black 6 mice. This is our study design at the top. Once the tumor is established and there's something to inject in the flank, we begin by oral gavage, treating them with the MEK inhibitor, and then they get the adapted T-VEC expressing the murine GM-CSF every 4 days at the arrows, and then we monitored them for growth. And again, what we saw was that combination therapy was associated with decreased tumor growth in this model and was associated with improvement in survival. And again, I just want to point out, I don't want to overread the mouse models, but about half of the mice really seemed to have improved survival and half did not. So we'll come back to making that better. In those mice that did reject their tumor, we did re-challenge them, and the majority of these mice remained tumor-free, suggesting that we had in fact developed memory responses in these animals. And this was associated with an increase in CD8 T cells that produce granzyme B. And this is just immunostaining showing the accumulation of the CD8 T cells that was increased with both T-VEC and MEK inhibitor treatments alone, but was greatly increased when you had the combination. This effect does depend on CD8 T cells, so we actually depleted CD4, CD8, and macrophages, and it's only when you deplete the CD8 T cells that we lost the effect. So the treatment is almost certainly dependent on CD8 T cells. And then we also became interested in whether these bad F3 dendritic cells may also be involved. So these are dendritic cells that are very important for presenting viral antigens and viral clearance. And recently, some of the work that Stephanie Spranger's done has shown that they're also critical for anti-tumor immunity. And what we found was that, in fact, in bad F3 knockout mice, that we do in fact lose the therapeutic effect, suggesting that both the bad F3 DCs and CD8 T cells are important for this effect. So the other thing that we took a look at is the antigen specificity of these T cells, and so not surprisingly, if you treat with T-VEC, we do see an increase in HSV1 glycoprotein B specific T cells, which are not really seen in the MEK inhibitor alone group, and these are further increased when you have the combination, suggesting that we are getting much stronger antiviral immune responses. And we looked at two different melanoma differentiation antigens, GP100 and TRP2. And while you do see an increase of these with monotherapy, the quantity of these T cells was significantly increased with combination therapy, suggesting that we were in fact seeing spread of the antiviral response to an anti-tumor response, consistent with antigen spreading and tumor regression. We also did gene expression analysis, and this is looking at the 18 pro-inflammatory interferon gamma gene signature panel that was really developed by Merck, that's shown a response, able to predict responses to pembrolizumab therapy. And we found that this was actually very good for also predicting responses to treatment. There's a bit of upregulation with T-VEC alone, not so much with the MEK inhibitor, but with the combination, almost all of the genes that are associated with interferon gamma inflammation were in fact greatly increased. And also the second gene at the top there is actually PD-L1, and suggested that we were upregulating PD-1 and PD-L1. So this is probably the biggest threat to effective oncolytic virus therapy, is that if you actually have a good type 1 interferon response, you will in fact upregulate PD-1 and PD-L1, and this should shut down the T cells. And this is just data showing that both the PD-1 and PD-L1 are increased with T-VEC alone, but are significantly increased with the combination that we have. So this really suggests that combining this with the PD-1 or PD-L1 blockade would be very effective. So if you remember, I showed you that we were able to treat about 50% of the animals. What happens when you add in an anti-PD-1? Well, this is what happens, is we then go to about 90% survival in these animals, suggesting that this 3-drug combination is going to be particularly effective. And so just to kind of, and the other thing is that the 3-drug regimen actually led to a further bump in the CD8 T cells from the combination alone, suggesting that we could further drive the immune response with this combination. So to kind of summarize, what we think is that when you have a cold tumor, which we would probably find would be generally low in sting, that the oncolytic virus, perhaps with a targeted therapy, and I would be very interested to look at other targeted therapies. For example, PARP inhibitors are now showing some really interesting activity in combination with immunotherapy and perhaps low doses of chemotherapy that can turn these into hot tumors. That alone may be enough in some patients, but in other patients you're going to have to really deal with the upregulation of immune checkpoints, and by adding these immune checkpoints in, you can potentially then get much better tumor clearance. And this is, of course, a hypothesis that's easily tested in melanoma, where essentially all 3 of these drugs are now individually approved for therapy, so we're very interested in pursuing that. So now I'm going to show you a little bit of clinical data that actually supports this hypothesis in patients. So this is a little-known study we published in which we did actually, as far as I know, the only randomized study to actually now look at an oncolytic virus plus immune checkpoint inhibitor. So this was looking at T-VEC plus ipilimumab versus ipilimumab alone in patients with metastatic melanoma. We published this in the JCO, and these are the waterfall plots. And essentially what we saw, this was 198 patients that were randomized in this study, is that the combination was associated with a 38% objective response rate versus 18% for IPI alone. And unlike in the OPTIM study, where there was only a 15% response rate of uninjected visceral lesions, in this particular study, visceral lesion response rate was 35%, which is, I think, really quite respectable compared to 14% for IPI alone. And another important point is, while we did see sort of typical ipilimumab toxicities and typical T-VEC toxicities, we really saw no additional toxicity from the combination. So these study patients, actually, we are awaiting the overall survival data, and I am told that that is now completed and is likely going to be presented at the ESMO meeting in the fall. Another little study that's gotten a lot of attention is a study that was published by Tony Rebus as the lead author, looking at pembrolizumab and T-VEC. And in this small phase 1 study of about 25 patients, a response rate of about 62% was seen, with about a third of the patients having complete responses of all injected and uninjected disease. And this is the swimmer's plot showing that essentially all the responding patients were still in response at the time of this publication in Cell. Based on this, the study was adapted to a randomized phase 3 study, and 730 patients have now completed enrollment of T-VEC plus Pembro versus Pembro alone, and that data is maturing as we speak, and we're all anxiously awaiting the results of this trial. What Tony did was some very interesting immunology in this, which again highlights this general hypothesis. So these were biopsies looking in green at CD8 T cells, and the red is PD-L1 expression, and confirming that in fact T-VEC was associated with upregulation of PD-L1 and recruitment of T cells in patients. And you can see here that even by week 6, there's this pretty significant influx, and this is persisting up to a month later. This is a really important graph that's sometimes a little difficult to explain. So what they did here is these are individual patients, and you can see their response rates. So the yellow are complete responders, and they're sorted here by CD8 density, which is the bar graph, and below you can see the top line is looking at PD-L1 expression, and then the interferon gamma gene signature score, which is based on that nanostring data that I showed you previously. And in general, we think that if you have high CD8s, if you have a high interferon gamma gene signature and high PD-L1 expression, that these are the patients that would respond to pembrolizumab. So in this study, if you look down at this where the X is, you can see that these were patients who have very low CD8 T cells in their tumor. Many of these have a negative interferon gamma gene signature, as would be expected with the low CD8, and there's one patient there that doesn't have any PD-L1 expression, and yet all of these patients are having complete responses. So again, this is really is at least a little bit of patient data confirming the hypothesis that the oncolytic virus may be able to really effectively take these kind of cold, immune unresponsive tumors and turn them into better responders to immunotherapy. So I'm going to finish up by just talking about some of the things we're doing at Replimune. So this is a company that was restarted by Rob Kaufman to really kind of expand the HSV1 oncolytic platform. And this is similar but somewhat different from what was done with T-VEC in a number of ways. So first, rather than taking any old strain of HSV1, Rob set out and did a study, IRB-approved study, where they collected cold sore swabs of HSV from 181 patients, and then established these viruses in the lab to look at their oncolytic capabilities. And it was interesting in that there was about a 1 to 2 log difference between the various strains that came out of this screen. And so we selected a strain that was oncolytic in the most number of tumor cells and really had a higher potency in terms of the oncolysis, which I'm not going to show you, that was used to further develop this particular platform. A second modification is that Rob included the gibbon ape leukemia virus, which is a fusogenic glycoprotein. So this causes syncytia formation in infected cells and is predicted to increase the immunogenic cell death of the tumor cells, and I'll show you some data in a minute that supports this. And then finally, it is a platform to encode various transgenes, and we do have a pipeline where we started with GM-CSF since it's previously shown clinical validation, but we're now putting other immune genes, and I'll show you some of the preclinical data with some of the agents that we've selected. And we believe that we can probably effectively get about 5 to 6 different transgenes into the virus with stable replication. So this is some in vitro data. This was actually done in Kevin Harrington's lab in London. And this is just showing that when the GALV gene is actually present in the HSV compared to GALV-negative viruses, that we see an increase in immunogenic cell death as shown by release of ATP and HMGB1 and also exposure of ectocalreticulin. We've now done a number of rodent studies. So this is xenograft studies using the A549 human lung carcinoma line and MDA-MB231 human breast cancer cell line. And so what you can see is that if you inject it with vehicle, these tumors just will grow. And if you give it an HSV-1 virus alone, it actually has some effect on this. But if you express the GALV, you can really improve upon the tumor regression that is seen. The PIT1, which is the receptor for GALV, is actually not present in mice, which makes murine studies difficult. So this is actually a rat glioblastoma model in which rats are injected with tumor on bilateral flanks, and only the right flank is injected. And what you can see is that even with fairly large tumors here, that you see significant regression, and that even the uninjected lesions are regressing as well. We've also shown that the RP1 virus can induce CD8 T cell recruitment. So this is actually a control tumor that was injected with just vehicle, and so your eyes aren't going bad. There's no T cells that come into these tumors. And again, the top row is looking at injected tumors, and the bottom row is looking at contralateral uninjected tumors. And then in the virus injected, you can see here the red is CD8, that there is big influx of CD8 into the injected tumors, and there's also CD8 T cells that circulate into the uninjected tumors as well. And so right now the pipeline that we have is that our first virus, which is called RP1, encodes the GALV gene as well as GM-CSF. That is now in phase 1. The phase 1 study is accruing, and we're hoping to present the results of the phase 1 data at a meeting in the fall. And the plan will be to rapidly expand this into a multi-cohort phase 2 study in patients who've had tumors that have been somewhat responsive to anti-PD-1. So this will include melanoma, non-melanoma skin cancers, most notably cutaneous squamous cell carcinoma, bladder cancer, and MSI high solid tumors. And we've also entered into a collaboration with Regeneron, where we'll be testing the RP1 in combination with their anti-PD-1 cemiplimab in patients with advanced cutaneous squamous cell carcinoma. Our RP2 virus has been modified by including a anti-CTLA-4 antibody, which is sort of based off the ipilimumab backbone, and our RP3 vector, which is also under development right now, will encode the GALV, the anti-CTLA-4, and 2 co-stimulatory molecules, CD40 ligand and 4-1BB ligand. And the reason we're excited about that is the animal data shown here. So on what I think is your left is the RP1 virus. This is looking at the A20 murine lymphoma model, which is one of the few models that is sensitive to HSV infection. And on the right, you can see that injected tumors, while they do decrease, and there's some delayed growth in the contralateral tumors, you can see that when we use the RP2 virus, where local anti-CTLA-4 is produced, there's almost uniform regression of, there's one, the yellow bar is the one mouse that outgrew a little bit at the injected site and the uninjected site, but almost all of the other animals had complete regression of both tumors. And our RP3, you can see at the bottom, this is looking at just single murine CD40 ligand and murine 4-1BB ligand, also seem to have very good activity. And so we are now working on putting all of these in the same vector, and those preclinical studies are currently being completed as we speak. So to conclude, T-VEC is the first oncolytic virus that's been approved for melanoma. It was really based on improvement in overall an objective response rate and durable response rate, as well as progression-free survival. But I think now with the 5-year follow-up, we now know that there is also a significant improvement in overall survival. And this has been associated with a very tolerable safety profile. Our preclinical data suggests that MEK inhibition can enhance T-VEC anti-tumor activity in mice, and this may be further promoted by PD-1 blockade, and something that we're very interested in moving into the clinical setting. I think I've also shown you some data that the STING status may be particularly important for allowing effective T-VEC or other HSV and perhaps other DNA viral replication And I think the other implication of that, of course, is that perhaps STING status is a biomarker that might help us better select patients who might respond to DNA oncolytic viruses. And the work at Replimune is we do have a different backbone where we are expressing new genes such as GALV, which I think we've shown enhances immunogenic cell death and appears to promote better therapeutic responses in rodent models, and clinical trials have been initiated now in patients with a variety of cancers. With that, I'd just like to acknowledge my colleagues who help with the T-VEC clinical trials, and Jen Gansert and the people at Amgen who I worked with, and Rob Kaufman is my new boss at Replimune. And then finally my academic group at Rutgers and my new family at Mass General. And I have to say that I was lucky enough to get 6 months to do a sabbatical in Sam Rapkin's lab, who's here today. And a lot of the ideas that I showed you today really came out of discussions with Sam and his really outstanding laboratory group. Thank you very much.
Question?
Howard, it's a great talk. The MEK activity, this is basically on the tumor cell, or you think it's impacting the immune system? And is it only related to melanoma?
Yeah. It's a good question. So I think the MEK was predominantly on the tumor cell. I mean, it may be having some immune effects, but I think this activity is really related to what's going on inside the tumor cell. And what I didn't show you is we actually have done this in the CT26 model, and it worked even better. So when we add the MEK inhibitor to the oncolytic virus alone, we got 100% clearance of tumors. In fact, it was so powerful that we really had to lower the doses to almost very low subtherapeutic doses. So I think it may be applicable in other tumors, at least maybe in colorectal cancer. So I think that's something that we have to explore a bit more.
That's great talk. So my question is, how your local oncolytic virus expressing CTLA-4 blocker can also change the another untreated tumor immune phenotype? So my understanding is that CTLA-4 systemic infusion can remove such a Treg or suppressing T-cell suppressive effects. That's why I'm a bit wondering, your local treatment with CTLA-4 blocking antibody expression can control the untreated tumor. So do you have any idea about such a mechanism?
So if your question is, do we know how the local CTLA-4 is working? We don't just yet. But part of we had sort of two general thoughts about that. So one is that we think CTLA-4 is probably going to be very important at the time of antigen priming, so that if part of the mechanism is that the oncolytic virus is going to kill the tumor cell, release soluble antigens, and now you want to cross-present these antigens, this is probably going to happen, we now think, through these bad F3 dendritic cells, probably at regionally draining lymph nodes. This is where having CTLA-4 blocked might be particularly helpful at getting those CD8 T cells primed. At the end organ, which is going to be at all the distant metastatic sites, now you're going to have to deal with the problems of PD-1 and PD-L1, and that's where we do believe that we're going to combine this with systemic anti-PD-1. And the other rationale is that the toxicity of systemic anti-CTLA-4 is probably greater than anti-PD-1, so expressing it locally, we may be able to get around that toxicity and then allow the patients to get systemic anti-PD-1 and to really push sort of what we see with Ipi-Nivo responses in melanoma patients, but without the systemic toxicity of it.
Nice talk. I guess it's working. So in case of cold tumors versus hot tumors, cold tumors are usually depleted from T cells compared to hot tumors anyway. So basically, follow up on the first question, do you really think the oncolytic virus is working on the T cells within the tumor, or is it somehow causing the recruitment of T cells into the tumor?
So the question is, is it the T cells in the tumor microenvironment, or are we recruiting new T cells? And so we don't have a complete understanding of that. I think what our data shows is that you do get antiviral T cells initially, and then we are seeing these melanoma-specific T cells show up a little bit later. So I suspect the answer is going to be both things may be happening. So if you have a hot tumor and there's already T cells there, and now they're going to be suppressed, part of the way the oncolytic viruses may work, and certainly in the presence of anti-PD-1, you may be able to rejuvenate those, or you're now bringing new antiviral T cells in, which by themselves may be responding to the virus, but because you're getting essentially a bystander killing effect, you're now able to get cross-presentation to other antigens. That's the hypothesis. But in the cold tumors, I think you've got to start that process again. And so we haven't looked yet at the neoantigens. I know everybody's interested in that, so it's on the plans to do down the line. So I think it'll be important, and for all of us as we go forward with this in the field, to really take a look at the antigen specificity of these T cells to understand that better.
Thanks.
Thanks so much, Howard. Maybe one more question. I know we're running behind schedule, but while we are still trying to pull up the next speaker slide, one more mechanistic question for you. While you were probably not surprised to find MEK inhibition rendering tumor cells more susceptible to any sort of second challenge, were you surprised that it increases viral replication? And why do you think this is different from many other viruses where MEK inhibition reduces viral replication?
So I was very surprised because I actually predicted that we would see the opposite. Because of the apoptotic effect of the MEK inhibitor, I just thought the cells wouldn't live long enough to support viral replication, and we would see a decrease. So it was a completely paradoxic response, and I don't fully understand it. I'm a little bit intrigued by the ability of MEK, though, to alter the cGAS STING pathway. And I have a feeling that part of the biology is related to altering the oncogenic signaling pathways within the cell. Now, how that is leading to enhanced viral replication, I just don't know, but we've repeated it a lot of times.
It seems very significant increase.
And whether this is specific to HSV in some way or whether this will be true for I think maybe some of the other DNA viruses, this might be true as something that we've yet to explore.